Welcome. Today we have Karl Bacos, PhD, coming from Lund University Diabetes Center in Sweden, where he's an associate professor and associate researcher, assistant researcher in diabetes and epigenetics, and a researcher in ExoDiab, Excellence of Diabetes Research in Sweden. In his research, he's characterizing how epigenetic changes, i.e. changes in our DNA that alter gene activity without changing the DNA sequences, regulate the insulin-producing beta cells, and contribute to the development of type 2 diabetes. The recent paper that he and his group put out in 2023 was type 2 diabetes candidate genes, including PAX5, cause impaired insulin secretion in human pancreatic islets. And today, the title of his talk is PAX5, a new player in T2, type 2 diabetes pathophysiology. Welcome, Karl. Thank you very much for joining us. And yeah, I would love to hear more about your work.
Thank you. Thank you for having me. It's a pleasure to be here. Yeah, so I will basically just dive into the paper, which was published February 15th. So if you want to read the full paper, it's available. And I should say that this is a collaboration between several groups within the LUDC. So it's by no means my own work. It's a lot of people involved in this project.
Yeah, well, there's a lot of interesting work going on at LUDC right now, and a lot of excellent science pertaining to type 1 and 2 diabetes.
Definitely, yeah. And so this paper, in this paper, we did a transcriptomic analysis of human pancreatic islets, and we do this to identify genes that are differentially expressed in islets from donors with type 2 diabetes compared to islets from non-diabetic donors. And these genes then may be part of the T2D pathophysiology. And you can do transcriptomic analysis in different ways. You can do the bulk sequencing, which is basically just analyzing the whole islets, or you can do single-cell RNA sequencing. Bulk sequencing is good because it's cheap, relatively cheap, but there's no resolution. While single-cell sequencing gives you high resolution, but it's also extremely expensive. And we chose to do bulk RNA sequencing to take advantage of a very large cohort of human islets that we have access to. So this has been done before. So the question is, why are we doing this again? The problem with the previous studies is that there's very limited validation, both regarding to the identified genes. So the overlap between the different studies is very small, which is mainly due to very low power for many of these studies, especially for the single cell studies, considering, which is not surprising considering the price. And there's also no functional validation. So in these papers, they have basically haven't done any functional follow-up to actually test if these genes are important for type 2 diabetes. So the LUDC has collected islets for many years now. We collaborate with the Nordic Network for Islet Transplantation. So if they can't transplant islets, we get leftovers, and we use that for our research. And today,
can I interrupt you just for a quick second?
Of course.
For the Nordic Network for Islet Transplantation, what % of the islets are coming from those with type 1, or if any?
We don't receive any from type 1.
Okay, I just wanted to check on that. It's all type 2.
Yeah.
Okay, great. Thanks.
And non-diabetics, of course.
Right. Thanks.
So this is now one of the largest such cohorts that at least we know of. And our aim was basically to use this to identify novel type 2 diabetes candidate genes. And this figure shows you a sort of description of the cohort and of our analysis. So when we started, we had just over 300 islet samples, or samples from islet donors. We did RNA sequencing on 283 of these. We excluded some where the RNA sequencing data didn't pass our QC, and we also excluded islet preparations with a purity less than 5%, and islets that had been in culture for more than 7 days, which left then 226 data on 226 islet preparations. We further excluded donors with HbA1c in the sort of pre-diabetic range. And we excluded donors that had previous gestational diabetes and non-diabetics younger than 40 years of age, because none of the diabetics were that young. And in the end, we compared 138 non-diabetics and 33 people with diagnosed type 2 diabetes. And we call this the LUDC islet case control cohort in the next few slides. So we used DSEC2 to identify differentially expressed genes, and we corrected the analysis for age, sex, islet purity, and days in culture. And on the left of the slide, you see the overlap with the previous studies, both the bulk and the single cell. And our cohort is the one in green. And again, you can see that the overlap is very small, even though we now have increased power in our study. The overlap between the other studies is extremely small. For example, there are 0 studies common for all 5 bulk RNA sequencing studies, and there's 0 genes common when you compare our data set to the published single cell data sets. So again, highlighting the importance of, well, not the importance, but the effect of the method used and the analysis pipeline used and the covariates used and so on. Anyway, when comparing to these studies, we found that of our 395 genes, 94 have previously been identified, while 301 were novel. And I put novel within quotes because they're novel in the sense that they were not identified by these specific studies, but some of the genes have been identified in more targeted analysis. And on the right, you see a few examples of these genes. For example, you have the glycine receptor, which you know is important for beta cell function. You have the IAPP, which forms amyloid plaques in type 2 diabetes. You have P10, which is an important player in cell biology. And there's also a lot of interesting genes which we know affect beta cells among the ones we call novel. We characterized the differentially expressed genes a little bit. So we used sorted alpha and beta cells from our human islets and did RNA sequencing on those to see what cell types express the DEGs. And we found that the majority of the 395 DEGs are expressed in alpha and or beta cells in human islets. We also did gene ontology enrichment analysis on the differentially expressed genes to see if there were any important biological pathways affected. And as you can see, hormone secretion, which of course is important in human islets, were among the enriched pathways. We also looked at published EQTL data. So that's SNPs that associate with gene expression. And this is data from the INSPIRE consortium. And we found that there are 148 EQTLs that associate with expression of 120 of our differentially expressed genes. And 2 of these SNPs also associate with type 2 diabetes risk, 2 with glucose measurements, and a few other with other metabolic parameters. We also looked at published genome-wide association studies to see if there are any SNPs that associate with type 2 diabetes or glycemic traits that are located in the genes we find. And there are 149 SNPs located in 106 differentially expressed genes that associate with type 2 diabetes or HbA1c or fasting glucose and so on. We also looked at published MQTL data, so that's SNPs that associate with DNA methylation. And we found 490 SNPs associated with 176 methylation sites in 90 of our differentially expressed genes. And finally, we also looked at DNA methylation data from a case control study. And there are 732 differentially methylated regions in 262 of our DEGs. So there seems to be some epigenetic and genetic regulation of these genes that might explain why they are deregulated in type 2 diabetes. This is a busy slide, but I will try to explain. This is data from the International Mouse Phenotyping Consortium. We wanted to see if the genes we identified have been found to be important for regulating in vivo metabolism. And this database collects data on knockout mice. And of our 395, the 395 differentially expressed genes, there were knockout strains available for 168, and 125 of these were phenotyped. And there was data available for insulin levels or data from intraperitoneal glucose tolerance tests or DEXA scans for quite a few of these strains. And the bottom part is busy, but I will just very briefly walk you through it. For example, SLC2A2, which the gene I'm pointing at with my pointer, encodes a glucose transporter, which is important for glucose uptake in rodent beta cells. And this is decreased in type 2 diabetes. And knockout mice for this gene has increased fasting glucose. So that's how this figure works, basically. The take-home message is that many of the genes we find differentially expressed in type 2 diabetes have been found to affect in vivo metabolism in mice. So that's the take-home message, basically.
Yeah, there's a lot of information on that slide.
Yeah. So
you can sit with it for a while.
Yeah, it's in the paper. So if people need more time to look at it, they can go to the paper.
Yeah, no, that's a great tip.
Yeah. So the question is, have you found any novel type 2 diabetes candidate genes? So we selected genes for functional validation, and we start with our 395 genes. We required a fold change of at least 2, either up or down, which left 80 genes. We also required an association with HbA1c in donors not diagnosed with diabetics, meaning that these changes might predispose to diabetes, so they occur before you actually get diabetes. So they might predispose to disease, or they might cause the diabetes. So this left 31 genes, and from these we selected the top 9 genes based on Q value, and we also selected CHL1. Top 9 genes that have not been analyzed in beta cells before. And we also included CHL1, which has been shown to affect beta cell function as sort of a positive control. And also SLC2A2 that I talked about before, which encodes GLUT2, a glucose transporter. In rodents, it's the main beta cell glucose transporter, but in humans, it's sort of debated whether it's important in mature human beta cells or not. So that was also included in our selection. And on the bottom left here, you see the expression of these 11 genes in our islets. And yeah, they're all significant, of course. And on the right, you see that all but one of these genes have either a T2D DMR, islet EQTL, GWAS SNPs, or islet MQTLs associated to them. So explaining why they might be deregulated in type 2 diabetes. So CHL1, HHATL, OPRD1, and SLC2A2 were all downregulated in type 2 diabetes. So we mimic this by using siRNA-mediated knockdown in human islets. And the middle panel shows you the expression, qPCR, of these 4 genes after knockdown. You can see we significantly lower expression. And on the right, you see insulin secretion data from static incubations. And you can see that islets with knockdown of OPRD1 and SLC2A2 have reduced glucose-stimulated insulin secretion, so the pinkish bars. While islets with knockdown for CHL1 had reduced basal secretion, so secretion at 2.8 millimolar of glucose. So SLC2A2, the GLUT2, also has an effect in mature human beta cells, basically, is the main take-home message here. The other 7 genes are upregulated in type 2 diabetes, and we used lentaviruses to overexpress only 5 of them in clonal beta cells, We didn't do all of them. I don't have time to go into why. It's in the paper if you want to find out why. But we overexpress these 5 genes or proteins in Swan cells. So BARX1, NEFL1, PAX5, PCOSA2, and SFRP1. And again, we see effects on insulin secretion. So PAX5 have overexpressing cells have increased basal secretion and reduced glucose-stimulated insulin secretion. There are also some effects in cells overexpressing NEFL and PCOSA2, but PAX5 showed the strongest effects. There were also effects on cell number, here measured by its sort of total protein in the wells, which was significantly reduced in the PAX5 overexpressing cells. There were also some slight effects of the PCOSA2 overexpression, but again, PAX5 showed the biggest effect. So we selected this for future follow-up to do some mechanistic studies as well. But first, we used immunostaining of human pancreatic sections to see what cell type the PAX5 is expressed in. And you can see it. Non-diabetics, they have basically no PAX5 staining in islets, while diabetics have quite a bit. And most of it is co-localized with insulin. So PAX5 is mainly upregulated in the beta cells in human islets in diabetics. So the question then is, how does PAX5 perturb insulin secretion? And before I talk about that, I have to mention how insulin secretion is regulated. So glucose is taken up by glucose transporters. It's metabolized via glycolysis, and then pyruvate is formed. Pyruvate enters the mitochondria, where it's and the TCA cycle, and then that fuels the electron transport chain, resulting in production of ATP. This yields an increase in the ATP-ADP ratio, which closes the KATP channels. This leads to a membrane depolarization and opening of the voltage-gated calcium channels, and calcium rushes into the cell. And this calcium then triggers insulin secretion. And then there are also amplifying factors as well. But basically, mitochondrial metabolism yields ATP, which closes the KATP channel. That's important for the next part. Because you can treat cells with high potassium to depolarize the cell without having this ATP. So by treating the cells with high potassium, you can find out if the defect occurs before or after closure of the KATP channels, basically. And that's what we did. We treated PAX5 overexpressing cells with high glucose or with high potassium, and you can see that again, glucose-stimulated insulin secretion is reduced, while the potassium-stimulated secretion is slightly increased, suggesting then that the defect occurs before closure of the KATP channel, potentially in mitochondrial metabolism. So we next investigated the mitochondria, and we used the seahorse, which is a machine that we use to measure mitochondrial respiration. And here you can see the traces. In black, you have the GFP, so the control cells, and in pink, you have the PAX5 overexpressing cells. And you can see that there's reduced glucose-stimulated respiration, quantified in one of the graphs on the right, and there's also reduced maximum respiration. So the graphs on the right show that the difference between basal and glucose-stimulated respiration, and also the glucose-stimulated respiration in absolute values. Both are decreased. And this reduction in mitochondrial respiration was sort of resulted in reduced ATP production. So these traces are from Perceval expressing cells, and Perceval is a reporter that allows you to measure the ATP-ADP ratio. And as you can see at basal glucose, there's not much difference, but then when we add high glucose, the control cells give a nice increase in signal, while the PAX5 overexpressing cells do not. And that's quantified here on the right. So there's significantly less ATP in the PAX5 overexpressing cells. One potential cause of impaired mitochondrial function is impaired expression of the enzymes involved in TCA cycle or the electron transport chain. So we use Western blot to analyze this, and we analyzed the expression of citrate synthase, which is a TCA cycle protein, and that's reduced in PAX5 overexpressing cells. And we also use an antibody cocktail that detects one subunit of each of the electron transport complexes, and that shows that SDHB of complex 2 is significantly reduced, while a subunit of complex 3 is actually slightly increased. But these expression changes may partly explain the impaired mitochondrial function. As I said before, there was less total protein in the PAX5 overexpressing wells. So we used an MTT assays to verify the cell loss, and you can see there was a clear decrease in signal in the MTT assay in the PAX5 overexpressing cells. And this can be due to either increased cell death or apoptosis, or it can be due to reduced proliferation. So we analyzed both these parameters. A caspase 3 and 7 activity assay showed increased caspase activity, and that was also shown by Western blot for cleaved caspase 3, which is the active form, which was increased. So there's increased apoptosis. But there's also reduced EDU incorporation, which is a measure of proliferation. So PAX5 results in both increased apoptosis and less cell proliferation when overexpressed in beta cells. We also did transcriptomic analysis of these PAX5 overexpressing cells, and PAX5 is a transcription factor. So it's maybe not surprising that there were quite large changes in gene transcription. So there were more than 3,000 genes were differentially expressed in PAX5 overexpressing in swan cells. And this included 75 of the 395 DEGs that we identified, and 3 of the other genes that we selected for functional follow-up. So I show a few genes here. For example, there's SLC2A2, again, one of our other candidates, which was reduced in PAX5 overexpressing cells. And this is a logarithmic scale, so the changes are bigger than they look in this graph. And these 3,000 genes were also enriched for some very interesting cellular pathways, for example, insulin secretion, which of course is important in beta cells, glucose homeostasis, regulation of cell death, and so on.
What is this presynapse?
So it's basically, I mean, it's a pathway that's enriched.
A neuronal pathway, or the pathway directly affecting the beta cell?
So I can't remember the exact genes involved here, but I'm guessing it's the exocytosis machinery, which is quite similar in neurons and in beta cells.
Right, that's so interesting.
Yeah. And as I said, PAX5 is a transcription factor, so we wanted to see if it may explain some of the changes to the other genes that we identified. So we used pScan, which is a tool that allows you to check for transcription factor binding site enrichment. And 196 of our 395 differentially expressed genes have a putative PAX5 binding site in the promoter, and this is a significant enrichment. And we did a literature study on these 196 genes, and a quarter of them have been found either to affect beta cell function and or number, or have gene variants associated with type 2 diabetes or other metabolic traits. So these are also some important genes. We also did simple correlation between PAX5 and the other DEGs, and found that PAX5 correlates with expression of PAX5 correlates with expression of 126 of the other differentially expressed genes. And this includes 7 genes that we follow up functionally, again including SLC2A2. And finally, we also did weighted correlation network analysis, which allows you to identify expression networks. And this found that PAX5 is part of a network with 86 other genes. So basically, yeah, we try to, yeah, these data basically support that PAX5 may regulate some of the other differentially expressed genes in type 2 diabetes, and therefore maybe cause these genes to be dysregulated. So to summarize, we have identified almost 400 differentially expressed genes in islets from type 2 diabetics compared to islets from non-diabetic controls. Some of these expression changes may contribute to beta cell failure in type 2 diabetes. Our main candidate is PAX5, which is elevated in beta cells in type 2 diabetes. And this perturbs gene expression, mitochondrial metabolism, and insulin secretion. And PAX5 may deregulate many of the other differentially expressed genes. And this is the model figure we have put in the paper that sort of summarizes the paper in a visual way. And as I said, it's been published. It's in the February 15th issue of JCI. So those who want to read the paper can look it up there.
Yeah, well, it's a very, your methodology is really thorough. You know, it really makes the case that the same should also be done, the same sort of march through these data should be done for T1D. I haven't seen a paper like this before that goes so very thoroughly and kind of like grooms through all these different genetic interactions, as well as, you know, and identifies, you know, the one gene of interest in this region. So it's really great. And I would also say, you know, just to kind of like throw it out there, what, you know, what's the trigger in your mind, or do you have any ideas of what the trigger for this elevated PAX5 might be?
So if I go back to, hang on. There we go. Oh, no, I went too far. Oh, sorry.
Okay.
Oh, my computer is not happy. Yeah, so we looked at these EQ-tails, the MQ-tails, oh, come on. I'm sorry.
Don't worry.
Now it stopped. Now it should go back. Now I'll do it gently. There we go. So PAX5 is down here, and you can see it has an islet MQ-tail and a differentially methylated region. So there's differential DNA methylation also in type 2 diabetes. So we believe it might be epigenetically dysregulated in diabetes. And what causes the epigenetic changes, we don't know yet.
Right.
That's a good question.
Yeah, that's next. That's probably the next paper. So
could be.
Yeah. And yeah, I'm sorry if I interrupted you before you were going to say a shout out to your colleagues and contributors.
No worries.
But it's really it's a fascinating story, and you know, very, very fine, a fine dissection of this pathway, really.
Yeah, we wanted to dig deep. You can always dig deeper, of course, but you have to stop somewhere, right?
Yeah, that's correct. And it looks like you have a lot of collaborators and a lot of you know, excellent resources here. What's next for your lab, and are you looking for postdocs, or what's going on there?
So our center, we are doing a similar study to look at genes regulating insulin secretion, like in non-diabetics. And our lab, I work with Professor Charlotte Ling. Our lab specifically, like you said in the beginning, we focus on epigenetics. So we are continuing our epigenetic analysis of human islets, and that's what our focus is at the moment. We're not working on PAX5 right now, but maybe we will pick it up in the future.
Okay, well, that's fantastic. I'm opening it up to anyone who has a question. I see that we have somebody, but feel free to put your question in the chat, or you can unmute yourself. No questions today. And I guess if you have any further questions, you can reach out to Dr. Bacos at karl.bacos@med.lu.se. And I'd like to say thank you very much for joining us today and walking us through this very elegant paper. It was really a pleasure. And I guess
Thank you for having me.
I'll try my hand at this. Tack så mycket.
Varsågod.
Okay.
Yeah,
Thanks
that's you're
again.
welcome.
Thanks again. Bye-bye.
Bye.